Yeah.
Happy That's my Okay. Did you get your hair cut?
Nope.
Did you comb it differently?
Just combed it.
He's just gussied up today.
You look very professor today.
I am.
You always look very professor. Like a classic professor.
I'm professorial.
You are.
Show and tell, while we're getting ourselves together here. Evidently, there are a bunch of people that got caught somewhere in snow or something like that.
Oh my gosh, yeah, some places got snow.
Didn't snow my house.
I know, mine either.
I got a light testing.
I'm not gonna.
So what I was going to share with you real quickly, and then we'll see if you've got other stuff too, was that, and I just brought B North, but that'll do. The infection quarterly reports, do you guys see those?
No.
That come out of the hospital? We give them for both for Bethesda North and also for Good Sam. And I downloaded these real quickly, and I just pulled off Bethesda. But for instance, where was I looking at this before? The number of cases of Clostridium difficile, which nobody can pronounce, so they call it C. diff. Was down 33% over the last quarter. That's significant change. Significant change. Bless you. And the number of catheter-related urinary tract infections actually held steady. And that's the most common one. You know, that's a nosocomial infection. Somebody inserting a catheter poorly, give somebody a E. coli infection out of all of that. But I'll see if I can go back and get the Good Sam ones out of all of that as well. But the Clostridium difficile drop is significant. And they've been really working hard at that over the past 2 years to get those numbers down. So anyway, and then one other thing I got here is that this came off the BBC Health News. BBC stands for
British
Thank
Broadcasting.
you. Excellent, excellent. And let me just read part of this to you. Lab tests in mice, and again, lab tests in mice, you always got to be a little bit skeptical about, but it's always where research starts for mammalian kinds of studies. At Cornell Medical, revealed a toxin made by a rare strain of Clostridium perfringens, which is another bacterium very closely related to Clostridium difficile. This one causes something called gas gangrene and is also associated with food infection. And probably everybody in this room has had some kind of Clostridium infection at one time. You blamed it on the flu, but you poisoned yourself. So anyway, and this rare strain of Clostridium perfringens produces a multiple sclerosis-like damage in the brain. Earlier work by the same team identified the toxin-producing strain in young women with MS. But experts are urging caution at this point, and they say there's more research that's needed to make sure that there is this direct link now between the two of these. No one knows the exact cause of MS, but it's likely that it's a mixture of genetic and environmental factors, which play a role. Obviously, it's a neurological condition. So anyway, the gist of the whole thing is, and I won't read the rest to you, is that what they found is that a particular toxin produced by this particular bacteria, which is not uncommon in the gut, in the gastrointestinal tract, seems to produce these lesions in the brain that are just identical to MS. And it's been a question for a long time, what's the scoop with multiple sclerosis? What causes that? And why does it seem like there are clusters of MS that seem to be in certain areas, high incidence of MS in certain areas. So if they do link it back to bacteria, it would interesting way of kind of saying, at least we found somewhat of a link out of all of that. Anybody got anything else they heard, saw, read? Go with it.
This morning I saw a report on the news that said that there was a decrease in the amount of antibiotics used in children in the U.S.
Yeah, there's been a real pediatric researchers have stressed for 20 years to quit using amoxicillin, which is the drug of the diagnostically destitute. So when you don't know what to do and you want to appease a parent who's going, I'm tired of listening to my kids screaming all night long, we'll give you amoxicillin. And it is overprescribed for way too long. So there's an attempt to quit doing that. And so what they're saying is, the gist of that, and that's been out for a couple of weeks now, is that they slowly want to say, we're not going to start to administer antibiotics unless it's absolutely essential until the kid reaches the age of 12. Because we pack way too much antibiotics into young kids, way too much.
There was a study done at Children's talking about antibiotic resistance in children that we just saw they sent the employees something about.
Yeah, it's showing up everywhere. It's a major, major problem. Very good. Anybody else got anything? Go with it.
I work in a nursing home, and since I'm in nursing school, I'm always following like right up my nurse's butt. It's like, what are you doing? Can I watch? Can I help?
Yeah.
There's this one lady with a big wound on her bottom,
Yeah.
and I got to see her use
The cubitus ulcer.
What?
She's got a bed sore.
Yes.
Yeah, okay. Okay.
Anyway, I got to see him use a patch that was like a sulfa drug base. And I thought,
Right.
that was cool, so we just talked about sulfa.
Yeah. Yeah. Kind of looks like saran wrap. Yeah. Yeah.
That was pretty cool.
Yeah, it is. Why did she get that ulcer?
She's been in bed for 6 years.
Exactly, and not moved on a regular basis, and so on and so on. Yep. Yep. Okay, we'll keep those things coming. Let's review a little bit here in terms of what we did last time. You know, it's a week again that we were together last, and sometimes memory doesn't do us well. But we were talking about the characteristics of the prokaryotes, and more specifically, we're talking about the bacteria. And you can recall as well, hopefully, that when we were talking about classification, and we looked at Carl Woese's 3 domains, he said that we got the eukarya, the eukaryotes, and we're all one of those. And then we've got this prokaryotic group, but he didn't like to use that term. He simply said we have another domain down here, which is called the prokaryota, or the eubacteria. Really, that's the term he liked to use, the true bacteria. And then we have the archaea, or the archaeobacteria over here. So those 3 domains. So what we've been focusing on, starting with the last lecture, is this group down here, the eubacteria, EU meaning true, the bacteria, as a prokaryotic kind of an organism. And we talked about, in general, about structure. Now, what did we mention so far? That talked about cell walls. And so we saw that there were 2 basic forms of cell walls. And we're going to see that that's going to vary as we move through the course, but we talked about 2 basic forms which determine gram-positive or gram-negativeness of an organism. So it determines how an organism stains using a set group of stains. We talked about a little bit about cell membrane. We talked about the cytoplasm, and in the cytoplasm, what have we got? We don't have a true nucleus, we don't have any membrane-bound organelles. The only membrane we find is with the cytoplasmic membrane, but we find a cytoplasm which is packed full of ribosomes, which are these non-membranous organelles, these granular organelles. And we talked about some other characteristics. Some bacteria may produce endospores as a means of reproduction or as a means of survival. Survival, not as a means of reproduction, because one cell gives rise to one endospore. That's not reproduction. So it's simply survival. We said, and not all bacterium do that. Most typically, what group does that? Who forms endospores? be more, you're on your way. Gram-positive, and tell me more. Bacilli that live in the soil, exactly right. And then we talked about the fact that some bacteria are motile, and if they are motile by flagellum, we talked about what that flagellum looks like, and we talked about how the organism uses that flagellum to move, and we talked about runs and tumbles, etc., and all of that. And I think that's kind of where we left off, if I recall correctly.
We talked about that.
So let's move on. There's some other characteristics we need to deal with then with these particular organisms, the bacteria. We saw that we had a membrane, and we saw that we had, typically we find some kind of cell wall, and you're not confusing membrane with wall, correct? Right. Excellent. And we said there are always exceptions, that there are some bacteria that don't have a cell wall, but we said that most do. And we're also going to find, though, that most bacteria, if not all, produce around the outside of this, not part of the cell wall, but this, oftentimes a glycoprotein. What does that mean? Anytime you see the prefix gly, you know what? Sugars. And you know what a protein is. So it's a mixture of sugars and proteins of various kinds, and it varies between the species. But this gets laid down outside of the cell wall. If there are flagellum, they can kind of stick their way through that. And this goes by a variety of names. Sometimes the old term was a slime layer. Sometimes it's also called a glycocalyx. There's that glyco again. And a calyx, what does that word mean? Any clue? Sounds like helix, but John?
It's more like a cup, I guess. Like the calyces and
A calyx refers to a cloak, something that surrounds. So it's this slime layer to the outside, which varies. Sometimes bacteria, depending upon where they are and what's going on, will produce a lot of it and sometimes not. And you've all experienced this before, this stuff. So let me give you an example. Bologna's on sale. And your budget is such that bologna is going to be what's going to keep you alive for the next 2 weeks. And you go to the store wherever you happen to be shopping, and you realize that bologna is on sale. Great price. So you figure, heck, if I'm going to have bologna sandwiches for the next 2 to 3 weeks, I'm going to buy 3 pounds of it right now, and I'll have it. So great. So you have 3 pounds, and you find that it's already sliced, though, in the deli counter, which Kroger used to do all the time. You notice they don't do that much anymore now. They will cut you what you want. But they used to slice in advance all that stuff, and they stopped doing it for this particular reason. So it's already pre-sliced, and you buy 3 pounds, and you get through the first week, and you're doing fine. And you get about into the second week, and you notice when you open the package, you're making your sandwich in the morning, the piece of bologna kind of slides between your fingers.
That's gross.
And it's got this kind of a slick feel to it. And you're going, well, not sure about that. But then you go, but if I put mayonnaise on the kids' sandwiches and put it in there, they won't know the difference. And so you go ahead, and you don't eat it, but the kids do, and they come home, in the afternoon, going, I messed my pants. Halfway through the afternoon, I couldn't make it to the potty, and I messed my pants. And what that slickness was this capsule. I mean, we've all seen this. You've all, another example, a great example of that is you've got milk in the refrigerator that you've had maybe 2-3 weeks. It was hidden in the back. You didn't, and you decide to pour it out, and it comes out with bloop, bloop, and it's got these strings that bloop attached to it. That's all because the bacteria that are in there are producing these slime layers, or these calyxes. What?
Her stomach was hurting this morning.
And most times when your stomach hurts is that you ate something bad. And don't blame it on the flu. It's you ate something bad or somebody poisoned you somewhere down the line. It's typically what happens. So why a calyx, a glycocalyx? Why would a bacterium do this? You made it. Why? Why produce this? There's got to be a reason.
It's trying to protect itself
Okay, in what way?
from the environment around it.
Okay, be more specific than that.
To help them stick together.
Okay, one is that this acts like a sticky, gluey thing, and bacteria, we realize, don't exist, rarely did they exist as a single cell. Bacteria stick together in groups, and we call those groups what? Biofilms. So this allows for the formation of biofilms, which is the way most bacteria like to live. And not even necessarily all with their own kind, they will form biofilms with other species of bacteria. And as we move through the course, I'll give you examples of this, why sometimes it's very difficult to treat certain types of infections because of the biofilms that they form and things like that. So they stick together, so it's a means of that. If something is sticky and slimy, you don't want to eat it though, right? And so who's feeding on bacteria in your body or out in the environment? In your body, who comes around? You get an infection, these bacteria show up, they're eating and reproducing in this lesion on your butt. Who's going to show up? Wandering micro and macrophages, correct? Mm-hmm. And what do? They endocytose, but they don't like to endocytose things that are sticky and slimy. So it inhibits phagocytosis. So adhesion, it'll stick to given surfaces. It allows them to stick together to form biofilms, and it provides protection against phagocytosis and against antimicrobials. Chemicals can't get through that layer. So these are incredibly important for the bacteria. And treatment now is, for certain types of infections, involves breaking up those biofilms. We've got to somehow disrupt those in order to let other types of antimicrobials work. And as long as we're talking about those, some bacteria also, not all, but some, especially gram-negative rods, have these protein rods that stick out. Not flagellum, they don't move. Well, I shouldn't say that. They don't move like flagellum. But they are relatively firm protein rods that extend out from the cell, and they go by a variety of names. One of the perhaps most common name for these is pilus is a single term. Pili refers to them in plural. For instance, E. coli, our little friend Escherichia coli, of which we've got billions of these things right now sitting in our colon, all of us do. Under certain environmental conditions, E. coli will produce these. It doesn't make them all the time, but under certain conditions it will make these. If we were to take a cell right now from a stool sample out of your rectum, I could show you E. coli that have massive numbers of pili, massive numbers. And the question would be, why? These are, again, somewhat rigid rods. It makes the bacterial cell look like a porcupine. But why would a cell then produce these? There's got to be a reason. Think porcupine first off.
It's trying to protection.
From macrophages again, absolutely. So anything that's going to come down and try to feed on these has a difficult time, and they're kept away. So it's a means of protection from phagocytic cells that would gobble these things up. Another reason, think capsule, or think glycocalyx, which, by the way, let me just use this term as well. Slime layer, glycocalyx, capsule, another term for these.
Does it help it adhere to certain surfaces?
Exactly. So again, we find the ability to stick to something, to adhere to something. Now that something, and in the case of E. coli, is going to be the cells that line your colon. And what have we got in those cells? In the membrane, there are all kinds of protein receptors that are used for this, that, whatever they happen to be. But E. coli has the capability, here's your cell, with this protein kind of stuck in its membrane, and this allows the bacterium to stick to those cells. Now, if it's a pathogenic organism, being able to adhere to those cells initially means what can happen later on. Then we can go ahead and attack that cell in one form or another. So it's a means of adherence, and so it becomes then also then a virulence factor. We're going to talk about that word a lot. We mentioned it last time. What does that mean? If something is virulent, we know what that means. Okay, something that can cause disease, can be infected. So if you have virulence factors, these are any characteristics which allow you to be infective, right? To cause infection. So capsules would be one, because it allows you to form a biofilm, maybe on your teeth or your tongue. Pili be another virulence factor, wouldn't it? Because it allows you to attach. Keeps away phagocytes. What about cell wall? It's a virulence factor, isn't it be? And with gram-negatives especially, what did we talk about last time? What are some factors that, especially in gram-negative bacteria, associated with the cell wall that allows them to be virulent or infective? The presence of LPS, lipopolysaccharides embedded in their outer membrane. That's another virulence factor. So anything, any characteristic that an organism displays which allows it to be an infective agent and a better infective agent, we refer to as virulence factors. So hold on to that. Okay. What else can we talk about? I think we kind of hit most of the outside stuff, and there's a lot of factors involved. Interestingly enough, just a couple of years ago, I was at a meeting in Denver, and actually it was Fort Collins, and there was a gentleman who, and I can't remember where he was from, which university it was, but he had video of a gram-negative bacteria producing pili, and the pili, everybody was astounded by this, would attach to a cell, and the pili would shorten and pull the bacterium in. Nobody had ever seen this before, because we thought that pili were just stationary. But what it appeared on his video was that the pili actually shortened, like almost like actin-myosin kind of thing, and actually pulled this cell over to that. So there's more research going on in that. If pili are actually involved with some type of movement, minor movement about it, we don't know. Last thing, let's look internal to the bacterium. We know that we don't have membrane-bound organelles. We have loads of ribosomes. We don't have a nucleus, but that nuclear area is sometimes called a nucleoid. But what do we have DNA? Absolutely. When we think of your cells, we think of how many chromosomes? Your cells, 30.
32.
You sure? 46? Do we hear 48?
No.
Yeah, 46.
46, 23 pairs. When we talk about the bacterium, though, we use this term. They're haploid. What does that mean?
Double.
Haploid, as opposed to diploid. We are diploid.
Singles? We're pairs.
Which is paired, haploid or diploid?
Diploid.
Okay, diploid means you got a pair, which meant that you were the end result of a sexual union. And we all were, whether you like to think about it or not.
So it's asexual.
We all were.
reproduction?
So now we've got bacteria which are haploid.
So it's
So now this says something about their love life, doesn't it?
asexual?
Yeah, they are organisms which don't undergo some kind of sexual reproduction because they are haploid, which means they have how many chromosomes?
23.
Okay, no, they have one.
Oh.
They just have a single chromosome.
So how do they go about reproducing?
We'll need to talk about that, won't we?
Sorry.
That's quite all right, but you raised a good point. So inside here, what we've got is basically a single strand of DNA that's kind of rolled up. Remember that silly overhead I showed? It looked like a bowl of spaghetti with pasta in there? So what we have is a unique chromosome. And it's not linear even. You know, our chromosomes are linear. But what we have with the bacteria is a single chromosome, which is, envision this. What's the best way we can do that? I take a big ball of yarn. So I bring this ball of yarn in here, and I have one of you stand over there, and I have one of you stand over here, and I give each one of you the end of the ball of yarn. Okay, so now we have a single strand. It's running across the room. Each end, one there, and I ask you to approach each other, and as you come in, just kind of take the yarn and wad it up. So everybody's wadding it up, and we finally end up with this mass. But then I tell you to take the end, this end, and tie them together. So now we've got this wad, but it's actually in the form of a ring. So it's a single chromosome, which is really united at both ends. It's not linear, it's a ring, and it's all kind of jumbled up and lumped together in this wad in the middle of the cell. So it's a unique kind of a chromosomal formation. But what is the functional unit along a chromosome?
DNA.
What?
DNA is the chemical that makes it up, but what's the functional unit along a chromosome? Something called a gene. Yes, a gene. And we know how genes work.
One leg at a time.
It gets more confusing. And what we're going to see is that the basic way in which genes are read, we get information out of them, for bacteria is the same way that we do with ours. It's just that the DNA is set up in a different way. And there's only one of those chromosomes, major chromosomes. But then to confuse things a little bit more, is that what we find is that most bacteria, at least at some time, are going to have extra little rings of DNA. So while there may be a couple of thousand genes on this chromosome, they may have these extra little rings of DNA, which have 5, 6, 7 genes on them. And anybody know what these are called?
No.
I give you $1.
Like that's gonna help us find the answer.
I'll leave it out there and break. I'll see if anybody gets the $1. You got a better chance of getting this $1 than you do of winning the football game if you bet for Denver.
Sad game.
They're extra little rings of DNA. We'll see if you can come up with a name for those. And we'll talk more about those as we move later on in the course, because the funny thing about these is that sometimes some genes in these can jump over to that main chromosome and jump back over here, and we have things called jumping genes.
Genome?
Nope. Okay, so what we've done is kind of said these are general characteristics of bacteria. Again, I want you to keep in mind that not all bacteria share all of these characteristics. What would be the basic that every bacterium shares?
Is it a plasmid?
You are so smart.
Yay, get $1.
You do.
I'm broke, I need it.
Right. Now you get a little deadly.
They're called plasmids. Well done. You get your $1 at halftime.
House.
And they're, what are they?
We get a Bruno Mars.
Those are extra,
Genes?
they're little rings of DNA that may have only 4, 5, 6 genes on them. And again, the question you should be asking is, well, why? And as we move through the course, I think you'll be able to see, I'll give you some examples in terms of why some bacteria produce these. But all the possibilities we've been talking about here, what is the basics that every bacterium has? a cell membrane, right? Cell membrane, cytoplasm, a chromosome, a singular
Ribosomes.
chromosome, and ribosomes, and that's it, right? Because some cells have cell walls, some don't. Some are flagellated, some are not. Some produce pili, some don't. Do maybe they all produce a capsule? Let's hope, but we don't know for sure. So basically what we've done is have the basic model, and we've built on that and looked at all the possibilities. And these variations allow for us to do what? Because we take organisms that have similar characteristics and we tend to put them in groups.
Classification.
Yes, we're getting a classification. Because what determines your characteristics? Your DNA. So if we have similar characteristics, and this doesn't always hold water, because sometimes it doesn't work, but basically it does, and we have the same genetic makeup, we're going to put you in this pot, and put you in this pot, etc., all the way down the line. So it starts to come up with a means of classification. Okay, let's jump away from the bacteria for a while and talk a little bit about some of the other microbes.
I had a question.
Oh boy.
How are they able to produce energy without, we have
Exactly, no mitochondria. So the question, it's a valid question, it's a great question. And that is, how can you possibly produce ATP if you don't have mitochondria? And the answer to that is, and we'll talk very specifically about that, is that the same enzymes that are there, that are found in the mitochondria, but those that allow for us to produce ATP, to undergo respiration, but the same enzymes, but they've got to be associated with the membrane. So where are the enzymes in a bacterial cell that we would find on the inner membrane of our mitochondria? Where's the only membrane? The cell membrane. So we're going to find the same mitochondrial enzymes embedded in or along the membrane.
But they're not real
But
mitochondria.
they're,
They're just
They're
like.
not mitochondrial. No. You give me that look.
Well, because I'm trying to figure out what you're talking about.
Okay, let's do a drawing.
Do I need to know it for this test? Well, then let's move on.
I want you to know it to be an educated individual.
Oh, but you already said I don't even need it for my job.
I want you to be an educated individual.
Right.
I don't give a flaming fricadoo if you ever become a nurse.
Oh, I know.
I want you to be an educated individual that can talk intellectually with people beyond your status.
Okay.
Okay?
Don't say beyond your status.
I want you to move up in the world. That's what I'm getting at.
Okay, thank
Okay?
you. All right.
Because if people find that you can speak to them on their level, they're going to say, come up and join me.
Right.
Yeah.
Oh, I get you.
Okay. Bacterial cell.
Right.
Mitochondria. Remember mitochondria? They look like that?
Yeah. Always.
Where does energy production occur in a mitochondrial cell? Along the cristae, these membranes that line the inner side of that mitochondrial cell. That's where all the enzymes are that allow for electron transport chain, chemiosmosis, the whole schlemiel that allows for energy ATP production out of all of this. Krebs cycle, all of that stuff lies along those enzymes that control all of that along these inner membranes. We're taking our bacterial cell, which doesn't have these organelles, and we're going to take basically the same enzymes. Some of them are slightly different, but they do the same thing, and we're simply going to pop them along the inner side of the membrane. Now, your next question should be, because you are becoming an intellectual individual at this
Right.
point, I'm going to turn you from a sow's ear into a purse.
Okay.
okay? Is what's going on here, right?
Well, I mean, yeah. That's what you're explaining to me, what's going on here.
What's the ultimate goal?
The ultimate goal is to produce energy to, like, reproduce.
Right, so does it make any difference, is the question I'm gonna throw out to you right now.
Where the energy is.
Yeah,
No, as long as you have it.
as we've got these enzymes. But the other question I was gonna make is, what seems to be absolutely critical to allow for this to occur? These enzymes need to be where? Embedded in a
Membrane.
membrane, yeah.
Okay.
Hold on to that thought.
Okay. My
I'm
face
trying-
really
I'm
that bad?
You have this look of consternation.
Okay.
Show everybody. And you know, give me a month and you're gonna have one of these aha experiences. Okay? Aha. Okay? Okie doke. So just be patient. Okay, what about the other microbes? Let's talk somewhat briefly about those. We'll see how many we can get through today. Talking kind of in general terms. What do you want to deal with first?
What are our options?
One of our options.
What are the options here? Okay, when we first drew up that list way back first time we met together, we said these are typical microbes we study in this course. And we talked about
Fires,
viruses,
archaea,
the archaea, keep going,
Algae.
the algae, the fungi, the worms. Let's do fungi. Let's talk about fungi real quick. Oh, boy. Okay, the fungi. See if you remember from the overhead that I threw up for you, that thing talking about a variety of names for the kingdoms, Whitaker's kingdoms. And the fungi fell into what kingdom? Ooh.
I have a picture of it.
Oftentimes they're referred to as the mycota or the mycotina. And we have all these people come up with slight variations on that. But what I want you to focus on right now is this, the prefix of the word, myco. Okay? Hello?
So fungi goes in the mycota?
This is the
category?
mycota. The mycotina is a kingdom which of the fungi.
Okay.
We call them the fungi. But the correct name for them in the classification scheme is the mycota or the mycotina. And what I want you to focus on, though, is the prefix of that myco. So you've got this funny rash between your toes, and you go to the Kroger store, and you look up and down the shelf, and you see it's mycotin ointment, mycotin, something mycotin, so and so and so and so. And what you notice is there are all these antifungal agents that have what in their name somewhere?
Myco.
Myco, exactly right. So when you think of myco, initially you're going to think of fungi right off the bat.
Yep.
Don't forget that. Promise me, Steve?
yes,
Okay.
yes.
So what are the fungi? So we think fungi. Throw some words out for me here. Okay, so let's get that out. We have no clue what that means, but we're going to throw it out there. It's actually a culinary term. Meaning? Has to do with food, right? Keep going.
Like,
Is moss a type of fungi?
Nope, good guess.
like old food. It grows, gross.
Old boots?
Old food. Like
Old
it is
food.
rotten-
Food.
rotten mold.
Okay, mole. Oh, mole. Okay, let's get that one up.
Old boots.
Is there like a dog vomit fungi? Yeah, it's
You're thinking of a Grex from yeah, it's not.
okay.
Yeah,
Yeah. Okay, so you took a shower this morning, and hopefully you did.
Mildew?
Who?
Mildew?
Yeah, and you looked at your shower curtain, it had this black stuff on it, or you looked at the grout between the tile and it had this black stuff on it, and we called that
Mildew.
Mildew. And you didn't know what that word was until you bought the Fantastic in the
Yeast.
store and said it kills mildew.
Yeast.
And you go, oh, okay, yeah, yeasties. Keep going. Any other words you can throw out here? Nope. So the question is, well, what do all these words have to do with the fungi? And of these, words like mildew and mold and mushroom are layperson's terms that have been thrown out there which attempt to describe something that nobody knows what they are. Except for maybe mushrooms, right? You know what a mushroom is. You have it on your pizza. Something like that. And you know that it's kind of looks in a certain way, right? Mushroom. But the yeast, you can think of that in one of two ways, either bread making or vaginitis. Okay? And the question is then, how do we get those two to merge with one another?
I'd rather not.
Okay.
Keep
But
those
let's
separate.
try and see what we can do with all of this. So, what is the relationship between the mushrooms on your pizza and the mildew on your shower curtain?
They're visible.
Okay,
I feel like
sometimes, right?
I'm gonna
Obviously, a mushroom is big.
Like the condition needed to grow it or something.
Well, maybe. Now we're starting to think.
well, yeah, like wet,
they don't
moist
photosynthesize.
areas.
Okay, so now we're getting this thing out. They don't photosynthesize, and we knew that that was a problem Whitaker had because they were in the plant kingdom and they didn't photosynthesize, so that's not. Moisture, they seem to need moisture.
And dark.
Dark, maybe?
Sometimes.
Sometimes.
You raise a good point.
No, not necessarily.
And
Heat.
They need dry, moist.
okay, so we're kind of all over the place. And you raise a really good point about darkness. We're going to come to that when we deal with certain parts of it. So let's take all the fungi, any of the fungi in the world, and we know that some of them are pathogenic, correct? So we have this pathogenic group, those that cause disease, and we have those that are non-pathogenic. And the non-patho forms, what is their role? To the ecologist, they play an exceptionally important role. What would that be?
Decomposition.
Yeah, they're decomposers, which means what? They're saprophytes. That's another term that's sometimes used. They're saprophytic. Saprophytic. Who did that? Goodness. Sapro means what? The Isle of, in fact, I just heard in the Aegean Sea this morning there was one of the Greek isles had an earthquake. But one of those Greek isles out in the Aegean Sea is the Isle of Sapros. And not Sapphos, there's a Sapphos there too. Sapphos was the Isle of Lesbians, correct?
What?
Sure.
Yes.
Yes.
Sapros, the Isle of Sapros, what was the common name for that? The Isle of the Dead.
And it actually has.
Oh, it's
People-
called that because you don't need, it's not like living, it's like, it's because of the conditions. Like, I get it. And now, keep talking. Just it out.
She's really thinking back here.
Okay.
Sapros has no means of anything to live.
It's like, oh,
Yeah,
So saprophytic implies that your source of energy is going to come from things that are dead or dying. And what are you going to do with those things? You're going to decompose them and
use them.
recycle them out of all of this. So we got a whole bunch of fungi that play that very important role. Very important role. On the other hand, we've got this other group over here which are pathogenic. And you can think of a whole variety of diseases, and we'll talk about some of those yet this morning. So we'll hold off for that. But this is one way of looking at the fungi, pathogenic, non-pathogenic forms. Another way is this. When we think of the fungi, what happens, for instance, during the summer in Cincinnati? You get the weather report,
Mold counts.
the mold count. The mold count. And what is that? They'll say the mold count's exceptionally high, so those of you who have allergies, may want to take your antihistamine or whatever it is you take.
Keep your windows
Um,
closed.
What is the mold count? Tuberculosis.
Spores.
Okay, now we have to throw out another word, spores. So we've got now what? Spores. And how do they play into this picture? Well, what we're going to find is that spores do play an important role here because virtually all fungi produce some kind of spore. Now, there are spores in turn can be divided into two groups, sexual spores and asexual spores. So sexual spores, whatever these things spores are, which must be like what? Little cells that get given off, and sometimes they end up in the air and we call them what? Mold, and we can count them in a given liter of air and it's called a mold count. And we know if they're in the air, what are we going to do?
Sneeze.
Suck them up your nose and they're going to get stuck on your mucous membranes way up in your sinuses somewhere and they may cause irritation. But the point is, coming back to that then, if some of these spores are sexual, that must mean they take on the form of gametes. Right? So they have to be kind of egg or spermish, but they don't look that way. In fact, in many cases, you can't tell who's male and who's female, but if they get together, they like to conjoin. And then we got a whole group over here that are asexual, and what would be the function of them? Simply to spread yourself all over the place, right? So to produce lots of things. So one of the other questions we're going to answer then is, what does mushroom have to do with spore formation? Because what it is basically is a spore producing morphological structure of the fungi. That's the ultimate goal of a mushroom. Produce hundreds of thousands to millions to billions of spores. And yet, what do we do with them? We take them before they go into sporulation, before they have a chance to start producing spores, and we slop them up with garlic and butter, and we eat them. Okay? So let's put all of these pictures together. Let's see if we can do that. And we'll start here, and then we'll take a quick break. So with the fungi, and this is always difficult, such a diverse group of microbes, and yet some of them appear somewhat macroscopic out of all of this. So then you say, well, really, why do you call this a microbe if I can see it? And we'll see if we can answer that question. Or just don't answer the question, or ask it and I won't answer it. But fungi typically are going to fall into one of two groups. The yeasts are the first group. And for sake of brevity, let's call them this. And the filamentous fungi. So what are we doing? We're dividing this very large and diverse group into one of two categories based on morphology. Based on morphology. So when we say filamentous fungi, it somehow must mean that what? Under the microscope, when I look at these fungi, they look like filaments, which means strands or something like that, exactly. So we've got some kind of a strand formation involved in this case. The yeast, on the other hand, typically are, if you look at the yeasts and you look at them under the scope, they typically look like very big cockle cells, but they tend to be oblong, more shaped like footballs typically than nice round cockle forms. And if you can remember from the lab last week in that handout that I gave you, one of the things when we were trying to measure the size of bacterial cells on that sheet that I gave you, it showed a yeast cell on there. And the yeast cell compared to a bacteria, which is this little, the yeast cell is huge. So these are much larger than the bacterium. Some comments about yeast then, real quickly. We know that some of them are what? Pathogenic. They like mucous membranes. In fact, they in some cases even like dry skin. We've got non-pathogenic forms. The non-pathogenic forms, we like them for what reason? We use them to make bread. as a leavening agent, and we use them to make alcoholic beverages. So they're used in the beer and the wine industry, and actually the whole liquor industry out of all of this. So how do they do that? Let's just take a look at them real quickly and we'll take a break. What's the characteristic of the yeast? Pasteur found it out. They can ferment, exactly. So we've got yeast then which have the capability of fermentation or fermenting, and that means what? They're capable of taking a carbohydrate, such as sugar,
Or turning into like.
and in turn using that sugar as a source of energy, but not completely breaking down the sugar, not completely metabolizing it, but the sugar got converted into a smaller sugar, which we now call alcohol, ETOH, ethyl alcohol, and CO2. So how in the baking industry do we use? We don't care about the alcohol in the baking industry. We want the CO2. So you mix the yeast, that little packet, which has live cells in it that are simply coated with the material that keeps them from completely drying out. They're sort of in a state of suspended animation, but you rehydrate them as soon as you put them in with flour, sugar, a little bit of salt, a little bit of oil, and some water, and they start to reproduce very rapidly. They get caught up in this anaerobic situation, and they start to produce alcohol and CO2. And what happens to your dough? It rises. And you look at it, you see all these little pockets in there. Those are CO2 packets of respiratory gas that simply get caught in there. And they make the dough rise. Lots of ethanol gets produced, but where does it go? It's gone. It's gone. And when you bake it, it's all gone. So in that particular case, we're using the fermentation to simply get the CO2. In the beverage industry, we may want the CO2, but what we really want is the alcohol. And if we do, in the case of a wine, like champagne, we want the carbon dioxide to still be in there, and it becomes a sparkling wine. In the case of, what's the difference between beer and wine? Source of the sugar, the carbohydrate, exactly. So instead of using grapes, what are we using? Grains of some kind, rice, wheat, oats, whatever that, corn will do, anything will do. And in the case of good, honest to goodness beer, what's the source of the carbon dioxide? The yeast. Cheap beer, Old Milwaukee. What they do is they kick that stuff out so fast and they usually lose the CO2, so what do they have to pump the CO2 back in? That's why you drink a couple of cans of that stuff and you're kind of going because it's all that excess CO2 that's in there that you're trying to get rid of out of that stuff. And in the case of hard liquor, what goes on? Same process.
Distillation.
But yeah, what are you trying to do is increase the alcohol level by distilling off as much of that alcohol as you can to concentrate that alcohol out of that whole thing. And then if you're going to, however you're going to flavor that in oaken barrels or whatever it happens to be out of all that. Let's take a break here. We've gone for an hour. Let's get out there, run around for 15. We'll come back and we'll finish this conversation.
Sorry. I actually remembered that I left my house. We were, yeah.
So Antoinette, are you studying for this test tomorrow? How are you studying for this test tomorrow?
Like personal lectures and stuff. I get those medications.
Yeah, all my friends that did really well last semester, they told me that their secret is recording the lectures and not talking about them because with 300 they tell you and give you a lot of hints on to study while they're talking. So that's what I'm gonna do is I'm gonna invest in a recorder, a good one, not the cheap ass one I have on my phone. Listen to lectures.
Okay, I got one, but I'm at home right now, so I just use this one for him, but,
Where'd you get?
I got it at Best Buy.
Was it cheap or expensive?
You can get, they come in like different memories, so maybe they have like one of like 20, and I got like the $50 one. Cause they have like, I think 3 times as much memory. But, I mean, I still got stuff in nursing too on there, but I can take it off. So I'm gonna probably do that soon. I just forgot about it and just
Right.
rolled it over to start using it again.
Right.
Christina, what did you say they told you about lectures?
Just to record them because that helped them a lot. And like they passed it, they were one of the people that one of my friends passed it with me, which I got really rare last semester. So they said recording helped them a lot, and they never recorded for nursing too. But it'll help you. That's what I'm doing for like after this test. I'm going out buying a recorder.
I have one.
Because I've never thought so much.
I just don't understand, like, I have so much, I'm looking at it for days and I can't figure out what the hell.
What I'm making sure that I know, and this is just based off of like, oh, what's helped me out before, is like knowing nursing implications for different things. Because I feel as though they're gonna give us questions like, you work at a clinic, a patient calls you saying X, Y, and Z, what do you tell them? You tell them it's all right, you tell them go to your fucking doctor, you tell them, you know what I mean? So
You're the mom, you'll know the best.
that's what people were like, you know what, you know, it's just something, you know. So I'm like,
It's your channel, you'll know better than anybody
So
else.
much of it is like the OB stuff and all the and I feel like I haven't seen it in action, so I'm not understanding it.
everyone says that that's like supposed to blow over. So I don't know. Just be positive.
Then I probably just gonna be
You really like where it's calling me later, right?
Alright.
Can we not have that?
Then I realized that, okay.
He's okay.
Sleeping.
Better. It's just hard for me to keep him awake during the day. But,
Good.
right.
Balance.
Right. I don't get shit done. But you gotta do what you gotta do, you know what I mean?
Understand that. How long does he sleep at night? Let me see. I'm like 9:30 Does he go right back to sleep after he eats?
Not usually. That's when he really gets me, you know what I mean? Like, he'll sleep for a good 5 hours or 4 hours and then he'll want to stay awake and he'll want to look around and he'll want to, you know, like, right. So, it's difficult.
Your boyfriend help at night?
No, not really. But it's okay, because it helps a lot during the day. Okay. So like,
He's gonna take care of that.
right, I try to. Okay. Yeah, and he knows like when school like tonight, he usually knows. He knows. So he knows. But yeah, most of the time because like he works and pays all the bills and doesn't have to be that housewife. The house is a mess. So I'm like, right.
I guess I'm backwards. I wanna be the housewife.
See, you think you want to be the housewife until you're like, this really sucks. I mean, I take care of the kids, you gotta cook, you gotta clean.
Maybe, yeah.
And it's a lot. I mean, I guess now cooking school takes up so much
I
work.
love to eat.
But yeah, I'm not. That woman is not me. Housewife, just because I'm an asshole. So I'll be like, you never leave. She's like, I'm at work, and I'm like, oh shit, that's right. You know? I don't know my place. I need to work. I need to get back to work.
I missed last week if I read instead.
There's a quiz every week.
Oh, at the end. Yeah.
You might let you make them up.
Oh, okay. We didn't do it very much because we got no, like he's so sporadic and I mean, look at my notes. They're like everywhere.
Just sample. What do we need to know for tomorrow?
I'm concentrating mostly on the lab values and stuff like that, because I figure the rest of it's common sense, like nursing process. Well, yeah. I'm concentrating on the lab values and then like the reflexes and stuff like development and invert and stuff like that. I'm not able to do. But otherwise, like I
Like
need.
the Palmer and
Yeah. And like when
when
I
they're
made flashcards.
disappeared.
I have flashcards in my backpack.
And it's one of their pre-class assignments.
Yeah.
I think they said, use your pre-class assignments. That's basically the professor telling you what you need to know about that topic.
Right.
Alright, I'll look back.
What business? Oh, for all of them, we're talking about pre-class assignment before.
That's what I'm concentrating most on. Because it's unrealistic to try and
Memorize everything.
Because it's production 3. Not tomorrow. No, not tomorrow, because we have a test. And then we
It's just
leave.
reproduction 1 that's tomorrow.
Right.
Is
But
there
as far
classes
as
after that?
That's what I was asking. Is it a class at my
no, because I know they try to work in remote
I'm
learning.
staying after lab to study.
Anybody actually watch the
I'll probably stay after.
remote learning for whatever,
That day that I memorized.
technology, informatics, or whatever?
You did?
Is there a lot of information in it?
On there.
But I thought that somebody told me to get
My teacher.
I'm gonna watch it because
supposedly that's what somebody told me. I don't know. They could give us access to that. Like, you know, how you go in and it says the name of people who have came in recently. I guess they can see local documents.
well, that would make sense because the reason why they do remote learning is to keep up with our lecture times. You know what I mean? Because it's in the lecturing. We have to keep this on hours.
I would rather than just do it in class because I would forget.
I know.
They usually stay there for like a month and then I get to go. But I get to
Studied. I studied on
Do you want those
Saturday.
flashcards?
Oh, sure, I'll look at them.
and now we can get it here.
Looking over here.
These are just for the reflexes.
Maybe it's like ours. Something just in case.
Yeah, we do.
It's not that far. It's
Yeah, you just did
5 to 6
Yeah.
and all these principals are great.
Yeah, it's like usually a 2-minute ride to children. It's just safer for your car. We like car
And they're like,
we
you
all
want to say,
drive
listen,
together.
cool,
We like to
this
drive
part
car.
of it. It's like,
I mean, it's nice.
she was, they called
So like, you're just watching and you're like, wait. I never went over there. It does, but. Like, I get out of.
You know, it'll give you more time to save up money. So when you come around, you have any residents to take. So when it comes down to 400, you get really fun shit. And you know what you can do is you can get your hours done. Because, and I was thinking all the stuff because all my friends are 400 was like lighting and
Yeah, we going like that, like mom, baby, and then,
There we go.
as, I mean, like a special care will be, and then this next week we'll go to, labor delivery. So we kind of
That's everybody.
it's like whatever, yeah, they kind of hop, jump across like that, and I'll be in there.
what I'm gonna do is I have like
Taylor. Which professor you have?
I got Ryan.
Oh, okay, so we'll start, so you'll
at children's, children's for children.
first the mom, baby, and then special care, then
Go to daycare later those days, and then I'll do that like once a week during the summer.
if you want to. You gotta make that like drug thing.
My baby mom. You gotta go to a
Well, that can do it for so long.
Okay.
it's right. She's one of my additional
That's
It's not that hard. I gathered my stuff and then I showed it to her and then I sent her something, but she didn't send me anything back over the weekend because I wanted to see badly in case I wanted to present it this week. But now I'm scared because usually she'll get back right here. And I know she's out of town. She didn't let me anything back. I wonder if she received anything.
Well, like I said, my teacher sent me like how to look at it, but she hadn't sent me anything back about how I did it. So they all might be done in the
You do all your work because if something happens, then how do I know that you want information and then are they going to count against me if they did receive it? You know,
especially if something goes wrong with their email, and it says from your end that you got it sent. But I guess you just print off that you sent it, just in case. You know what I mean? To have proof of accuracy. If they said no, you didn't, and you're like, yeah, I did. Just print it off. Show that it was sent. I hate that too.
I just remember getting hit.
Like you said, what if, you know?
and so I've never trusted them. I mean, usually I like to give like a hard copy.
Yeah.
And I mean, my concept numbers is that they
Yeah. You have a lot of hard work. Hard not to got it.
But I know she got it, so I'm not gonna hurt her, but I didn't even know that she even looked over this medication sheet I made us.
Oh, yeah.
I had the ballistic.
She probably has.
Because I really want to be ready to try to give it to a patient this week, you know. Yeah, we got other
Which one? Oh,
whatever we did this week.
We only just make a black.
We went, nobody signed up.
Oh, it's Pikachu!
About like how your brain stops creating.
I was like trying to decide, should I take your
Right, and you probably were stupid.
Well, I was trying to figure out why you kept shaking your head.
No, for
And we had to like pour.
We have class after the test.
But she, yeah, everyone's set up. I feel like,
But people always post pictures of them studying. I'm like, bitch, you are not studying. You did this. You know what I mean? Like staged it, make it look nice. Just say I know I'm studying my ass off on Facebook, on Twitter. You know what I mean? I used to do that shit until I realized, like, oh, it's been 3 hours. I just been taking selfies.
I don't think anybody expected that.
Oh, I expected. I can't believe they wait. Did they check the email? You know what? I read it.
They just checked to see
As
that
long
you're
as it's filled out, because if you have one part of one question.
Like, do they make you do it?
No, they were ugly old
Wasted 15 minutes on election.
Well, I got there and recorded Bill, and I had to finish everything, so I walked right back out the door and followed all the links, but I threw
Some people burn me. How dare you get cute to go to school? They just get really cute to go to school, or like to sit around you absolutely nothing. And you ask them who you trying to impress, and they say nobody. I'm calling bullshit. Don't get cute to go to school. I was like, do you not know how to curl your hair? I was like, yeah, I know how to curl my hair. I'll save that for more time. I'm going to like you wear your Sunday's best. There's a difference. They're just putting on makeup, flipping their hair like fucking Barry Fawcett, wearing boots, lipstick and shit. Because you wear your cute little ponytail tucked in and your collar and your sweater. You look cute, but you don't have to wear a girl costume.
I didn't know I was going to do.
Some bitches know all
I'd
that.
rather just look at professional.
It's because I'm looking on social media. This girl was like, posting pictures of herself looking pretty. I'm like, bitch, ain't you about to take a picture?
I've never heard anyone, like I always heard on the news, on my social media, I've never heard anybody say anything in social media.
But then those people end up being like 30.
You know where that is? That island? That island?
That island,
And
yeah.
it's in the Cycladic Islands.
Yes.
And my husband and I have a house on fire.
My goodness.
And so my husband's a marine biologist.
Yeah.
And we dove near this island.
That's pretty incredible. Pretty incredible.
And so my husband wrote his PhD thesis on, I don't know how to, but he wrote his thesis on the sea and archaeology. Because in the archaeological museum in Athens, he was a part of a group that found one that owned the shipwrecks in the Aegean.
So that's pretty exciting. Yeah. Pretty exciting.
So yeah, I got my husband's he's always got something, and he showed me a single cell organism one time when we were diving, and he was pointing at it and everything, and I was like,
There's a lot to see, awful lot to see.
and well, now I'm a little bit more interested, but the Aegean is really a real neat sea to dive in. I've dove in the Med, the Persian, the Aegean.
All the above. about
You're running on the treadmill.
But I've never
Etruscans
done
and
anywhere in the
Fitchburg.
United States.
Yep. Go figure. You got places to go and see. Okay, let's pull ourselves back together here. This is where we left off before we broke there, and everybody got a chance to get some caffeine in their systems. Yes? We were talking about the fungi, and we said that from a morphological point of view, we can divide them into one of two groups. They could be the yeast or the filamentous fungi. From a physiological point of view or an ecological point of view, we could divide them into two groups. Pathogenic and non-pathogenic, which are the decomposers or saprophytes. So there are a variety of ways in which we can kind of move these organisms around, and that's why it gets a little bit confusing from time to time. So we talked about the yeast, and we said that they are typically single-celled. They're involved with the process of fermentation, even though they may be pathogenic, they still ferment. They can ferment, and we use them for industrial and for food purposes and all of that. I'll throw this out to you just because sometimes this can be confusing, and if anybody spends any time with the yeast, which you may or may not do, I don't know what you're going to do with your life, is that sometimes you'll notice that they, in a new culture, you will see what looks like this, and you'll go, gosh, it's a filament. But it's not, because yeast, if they're reproducing rapidly, typically what they do is undergo a process of mitosis, and one cell divides to become two, but they don't let go of each other, and they just keep sticking to each other because they're reproducing so fast, and it forms, that's called budding, 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 and they reproduce so rapidly it looks like a filament. So it's kind of a false filament. So I just don't want you to be confused by that. If you're looking through the scope someday and looking at yeast, and you go, Rashke said that these were single-celled, and I see what looks like filaments, what's going on here. And that's that budding process. Okay, let's look at this group. Huge group. Filamentous fungi. They get this name because under the microscope, you take a look at them, which is what Hooke did. Remember Hooke? Okay. He loved the fungi, and he found that these fungi under the microscope formed these filaments or threads or trichomes. There are all kinds of names. And in some cases, they were, you could see individual cells, but in other cases it looked like the filament just kind of went on going, and he couldn't find any cross walls between them. So we know that they're eukaryotic, correct? Yes? So they got to have a nucleus, a true nucleus, and they got all the rest of those organelles in there, but he found some that didn't show cross walls. And it's just a unique kind of a characteristic of certain types of filamentous fungi. We refer to them as being coenocytic or cenocytic. Doesn't look right, but it's close. And they form a syncytium in which there are no cross walls that form. And why does that bother you?
And then allow for individual cells, so how are they going to reproduce if they can't?
Okay, that could be part of it. What else bothers you?
Multiple DNA, not multiple kinds of DNA, but multiple nuclei.
Okay, and you usually think of the nuclei kind of doing what? Kind of being the controlling, for a given region. And then in a case like this, how does this cell know that this is my jurisdiction and I'm not going to go beyond that? And so it raises some interesting questions, and we haven't really worked our way through that as microbiologists. So we just kind of take that as an interesting kind of a situation. And somewhere down the line, they lost the ability to produce these cross walls, or these guys learned how to produce cross walls, whatever happened. So we find these kinds of things. And what happens under the microscope is you see these filaments, and they start to branch in some cases, whatever, and they form typically what we refer to as a mass. And then we have names for the individual threads, and we refer to those as hyphae. Hyphae, I don't know if you can see that over here. So as you look under the microscope and you see this tangled mass of filaments, An individual filament is called a hyphae.
Where is that at?
Just the individual filament,
Oh,
one of these is called a hyphae.
okay.
But when they break and branch and they keep reproducing and you finally end up with this mat, you've all seen that before. You got an orange that went wild on you and it's got this green fuzzy stuff all over it. Or bread, you know, too long there and you open it up and you're going to make a peanut butter sandwich and it's all fuzzy. So the fuzziness you see, if you were to look at individuals of those, that's a hyphae. An individual filament or a thread is called a hyphae. The mass of itself, the mass of the whole thing is called a mycelium. And you've all seen that. It's simply the cluster of all of those hyphae together. Now, let's do some examples. So in the fall or in the spring, when it's very moist outside, and mushrooms show up in your yard. There they are. So the question is, well, what are those, and from whence doth they come? And typically, if you look close enough, you'll find there's a pattern here. In that you will see, let me just erase this and work in the middle. That in most cases in your yard, when mushrooms begin to show up, is that they will form somewhat of a ring. It may be very irregular, but they kind of come up in somewhat of a ring. And you've got mushrooms here, and you got another one here, and another one here. And they used to refer to these as fairy rings. Because these little mushrooms, you know, you came out in the morning and you look down and there are the little mushrooms, they look like little stools. And so what you thought was that last night when you were sleeping, the fairies in your front yard all got together and sat down and told stories around in a circle. And that's kind of where this came from. It's called a fairy circle. But what's going on here? Well, if you were to dig underground, go down about an inch to a half an inch, you would see that there is this mycelium all over the place in your front yard. And it can be 5-6 feet across. It could be 100 feet across. They can be huge. And it's simply these threads that have grown. And around the edge of that, when the conditions are right, what will happen is that these hyphae virtually will come together and form this dense body that becomes visible, and that's the mushroom. Starts off as a button, pushes its way up through the soil, and what does it do? Then opens up into what we consider a typical mushroom. So here's the soil line. You got all this mycelium underneath here. It masses together, pushes its way up, and all a mushroom is then, if you were to put it under the microscope and tease it apart, you'd see that it's nothing more than a mycelium. Massive threads all kind of jammed together. That's all it is. And in turn, it matures, correct, to form this kind of thing. And underneath that, depending upon, and if you think of the mushrooms you would buy in the store, if you know, Caprinus or just those common little white mushrooms that have no flavor and no value, but if you turn them over, what do you notice? They have gills.
Yes.
They have those thread. And if you were to take a single gill and cut it out and put it on the microscope, looking down at it, here's the side of a gill, and it's got these little, you okay? Don't you die on me, okay? Is that you've got hyphae coming off, and these hyphae are pinching off spores.
Actively, like, they're continuing to do that.
Yes.
Okay, cool.
So, and if you didn't pick the mushroom, what would happen? The spores drop out the bottom of the cap. They get blown away by the wind or the water. They get passed all over the place. And they in turn then give rise to new mycelium. So typically a mushroom is a spore producing structure. That's all that it is. And typically the spores produced here are asexual spores. Not always, but typically they're asexual spores. Now there are other methods where spores can be produced that are sexual, and that gets very complex. And if you've looked at your text at all, you notice that what Bauman does is takes all the fungi and dumps them into groups, the Deuteromyces and so on. I'm not asking you to memorize those names. Everybody with me? These different groups of fungi which are separated from one another based on how they actually produce their spores. I'm not asking you to know those. Everybody clear?
Yes.
Okay. Those are big, long names. You don't have to know those. But the point being here then, what is the function of a mushroom? It's a spore-producing structure. That's all that it is. Of what nutritional value?
Does it even have any?
Minimal nutritional value. Minimal.
It's gonna add a bacteria, not bacteria, but
The
fungus.
The one thing is that in the cell walls of fungi, there's a material called chitin, which is not much different from actually the exoskeleton of shrimp and lobster and things like that. And if anything, that provides bulk because it's undigestible. But you'd have to eat a boatload of mushrooms to get the advantage of that. So what do mushrooms do basically? You get a couple of minerals out of them, but
Flavor.
it's insignificant. They soak up flavor.
Yeah, they taste like what you cook them with.
Exactly right. It's so good to have you in class.
I talk a lot.
I like it. I like it. Thank you very much. No, I'm serious. And that's exactly what they do. So typically, what do you do with mushrooms? Saute them in garlic and butter. Or whatever, herbs de Provence or something like that, because they soak up those flavors, and then they're simply carriers. They're basically carriers. So raw mushrooms in your salad
Ew.
makes no sense. Doesn't make any sense.
Make you feel fancy.
Right.
Yeah, that's probably it, but it doesn't make any sense.
So
Yeah.
why do people put mushrooms on stuff then? Like pizza, they would taste like pizza?
Well, if you're going to make pizza, if you're a pie person, I was going to say a pie man, but you'd be a pie person, and you're going to put mushrooms on, what you've typically done is either brined those mushrooms or you've soaked them in olive oil with garlic. So if your pizza person is just putting raw mushrooms on there, this guy or woman doesn't know what they're doing because they're misusing the food because it's a non-taste event. And it doesn't do anything but shows you mushrooms on your pizza. Yeah,
But then I have an allergy to mushrooms, so is that,
and it's probably the chitin
okay.
because it's a unique type of a carbohydrate. And some people just don't deal well with that at all. You have seafood allergies as well?
No, I can eat seafood, but
Oh.
every time I eat a mushroom, I break out.
Yeah. And sometimes that's psychosomatic. We got a neighbor that if she finds a mushroom, she goes into anaphylactic shock. She doesn't even have to eat one. She found one in her salad, and then she's going, and she spends the rest of the evening going. So whatever. But there's something there. It could very well be.
Do we have multinucleated muscle cells?
Sure. Your skeletal muscle is multinucleated.
Okay.
Right.
So that's
And-
not so unusual then.
No, it's not in that respect. And think of the function of skeletal muscle. How does that happen? Going back to A&P, this has nothing to do with this, but you raise a really good question. During fetal development, you probably talked about this already. What happens to muscle, skeletal muscle cells? You have all of these myoblasts that fuse together to form a very long spindle-shaped cell called a skeletal muscle cell. It's multinucleated. And maybe why? What is that cell packed with? Actin, myosin. Those are proteins. Troponin, tropomyosin, all of those. Proteins, massive quantities of protein. Who holds the code, the blueprint for making the protein? The DNA does, right? Okay? So the more DNA, then the more genes you can read, the more protein you can make.
Right.
So it makes sense.
So
And in the
purpose
case
for that,
here,
no one knows.
maybe. Although these are non-modal, so there's nothing there to, you wonder why. Just to wonder why, because they're not packed with protein like we would find with skeletal muscle cells. Okay. I was going to say something else really profound. I can't remember what it was. It'll come back to me. Handout. We love these. Oh, yeah.
So are fungi
No, not at all. Only certain kinds. Those coenocytic ones. Why does that not look right? Cenoacidic. Yeah. You raised the question before about darkness. The little white mushrooms, button mushrooms you buy in the store, everybody's bought those before, right? Where do they come from? Extras? They're grown, and if you look, most of them, interestingly enough, are grown in Pennsylvania, but there are more and more there are more and more farms that are showing up with these mushroom farms. But they used to raise them in the dark because they thought that there's something about sporulation that happens in the dark. And they had these big bins and these cinder block. If there are certain places in central Pennsylvania where they still grow them, and they're these long cinder block, no windows in them at all. And inside you go in, and it's horse manure or cow manure that they're growing. So sometimes you get the button in the store and you still see the brown stuff hanging off the bottom. That's manure. But it's sterilized. It's been sterilized. So it's high in nutrients. And what they do is they save spores all the time from one bat. They sprinkle the spores over there. They keep them nice and moist. They kept them in the dark. And what would happen is the mycelium would grow and the manure, and what comes propping up along the edge were these mushrooms. They clip those mushrooms, they put them in a package, and they sell them. And they came to realize, though, because other people started growing the mushrooms without darkness, and they got the same product. So darkness has nothing to do with. You used to think, because when did the mushrooms show up? In the morning, right? So what have they been doing all night long? In the dark. Okay? But now doesn't seem to be any light factor involved.
In my yard, we have like orange mushrooms.
Sure, there are a variety of different colors, and there is a species around here. Whatever you do, nobody's a mushroom picker, right?
It all depends.
You crazy? The only mushroom that I will pick and eat around here, the only one, is something called a puffball, which is nice and round. It is perfectly white. And I know because I'll show you some of these in the lab. But usually they stay relatively small, golf ball size to maybe tennis ball size. They're not, sometimes they get huge. They get as big as a soccer ball. There's a lot of nutrients in the soil. But before they go into sporulation, you can slice those up in little garlic. But others, I'm always leery of, because Coprinus is, there's an extremely deadly mushroom that usually comes up nice and white. And people associate white, I'll get into your question here, white with being safe. And these things will kill you. So, I mean, do you actually go out and pick?
Yeah, there's like, it's around Easter time, but it kind of looks like a brain, like a mushroom that comes up. What is it called?
Like a truffle?
It's called spongehead.
Yeah,
Oh, the
like a
spongehead, yeah.
thing,
And
like a
that's,
truffle, but,
and that one you can pretty well assure because it looks so goofy.
Yeah.
You can't confuse it with
You cannot
something else.
confuse it. I always heard if you blanch them and they turn yellow.
Wrong,
that they're,
wrong.
you know,
No.
I would never eat any
Yeah.
of those
But,
ones.
and there was I had students before who said that their parents pick them, and they always put a penny in the bottom of the pot when they blanch them, and if the penny turns a certain color, they know that they're poisonous. No, don't do that.
So I have a question.
I'm sure.
If they are always actively, you know what I mean, making filaments and you know what I mean, reproducing.
Yeah.
So how do you dry, you know how like with like drug shrooms, they're like dried shrooms?
Right.
How in fact do you do that if they're constantly trying to reproduce themselves and like
Hold
how
on
do
to
you
that.
get it dried?
Hold on to that. Hold on to that thought. I'll come back to that. Anyway, going back. There is a species of mushroom around here that usually is just a beautiful orange on top, or orange, and underneath is kind of reddish. It's a gorgeous mushroom, very common around here. I get them in my yard all the time. They're also extremely toxic. Yeah. Okay. And so to the point that even if you pick them with your hands, you wash your hands.
My dad just hit him with a shovel.
Are they hard?
No, in
Just
fact,
got rid of them.
they're fleshy.
We used to
Fleshy
take
eating?
these ones,
Fleshy.
they'd grow,
Fleshy.
Oh my
they'd
god.
grow in like little balls and they'd be like either this big or real small,
Yeah.
but when they dried out, you could throw them and they.
Yeah,
like a
that's
black
puffball.
pot-
Yeah, that's puffball. Yeah.
You'd eat those?
Yes, before they sporulate, they're great.
They're the funnest thing.
Yeah.
Throw at each other.
Yeah, exactly. Yeah,
Smoke bombs.
yeah, and that's exactly the way it goes. I got some in the lab. I'll show them to you in the lab. Okay, take a look at the handout. And what I've done, this is very similar to what you got in your textbook. I modified it just a little bit. But from a medical mycology point of view, we can take the fungi that cause disease and lump them into the categories that you see down the left side and running over onto the back. So they fall into categories: the superficial mycoses, the cutaneous, the subcutaneous, the systemic, and finally the opportunistics. Because they're kind of grouped on the back, and there are a whole bunch more than that. So let's walk through some of these real quickly. Again, disease is there on the left. The actual agent, that means the organism, genus and species name, is there down the middle column, and method of treatment is off to the right. And I have to tell you, method of treatment changes almost monthly, because there's always somebody who's coming out with a new antimycotic agent or a modification of a previous one with a different name. So it's really tough to say that this is up to date, because in 6 months it's out of date again. So the superficie, the piedras. This is where we've seen most of this so far, and is because of the influx of Central Americans and South Americans. This tends to be a very common fungi found growing on the hair. And we see more and more cases of that showing up in the tri-state, in fact, all around the Northeast, because of the influx of those particular individuals who bring it in. Grows in the hair, it forms these kind of granular formations on the hair itself, and most typical of individuals with very poor hygiene is what it amounts to. Sideriasis versicolor, this is again typically found on the fur or on dead skin. Malassezia furfur, neat name. Sad story that goes with this, though, is that not that many years ago, there was a situation in I have to go back, I've got the record somewhere, it was either in Kansas or Nebraska, where they had 5 or 7 neonates die from a lung infection. And they were trying to figure out what the heck was going on, and they finally brought an infectious team in from the CDC, and they determined that in the lungs of these neonates was this organism, Malassezia furfur. And not normally found as a lung disease, it's typically found on the hair. But, what have you got with a neonate? Reduced immune response out of all of this. And they couldn't figure out, they had all these cases, and they treated a bunch of them, they lost a bunch of children, and they finally kept looking and looking, and they took it back to a nurse who had dogs. And in turn, in the morning before she left, she wore her scrubs to work every day. She would scratch her dogs and pet them and leave, and she came in with all these spores on her scrubs. And she took them in, obviously into a region which was supposed and nothing's ever sterile, but a region which was considered to be safe. So she brought the spores in with her, quite unknowingly. So just be mindful. You know, the whole idea of, I see so many people wearing their scrubs into work, and I don't know where they work as part of this hospital, but you should never do that. You should always change into them when you get here. So but the other part of the story is now you've got an organism which does what? Typically associated with hair disease forming a lung disease because it's an opportunistic pathogen. The second group, the cutaneous mycoses. We call these commonly the dermatophytes. And you know what derma means. Skin. And phytos means plant. So in a sense, we're talking about a plant that grows in your skin, except that fungi aren't plants. Sometimes I like this word better, the keratinophilic fungi. So what's keratin? Okay, so it's this protein that gets produced in your skin. And if you have philios, loves that stuff. So you get fungi that like that. And all of these running down the left side then are tinnias. Now don't confuse Tinea barbae and Tinea capitis with genus and species. These are simply disease names. But if you kind of look at them, you can kind of figure out why. And there are more species or genera that I've got listed here that can fall into this category. But for instance, Tinea barbae comes from the fact that it was associated with barbers that would used to shave men, and little nicks, scratches, and using the same razor over and over and over again without properly cleaning. They would come down with a ringworm on the face. That's really what it amounts to. So these are all ringworms. Tinea capitis, where's the ringworm? On your head. Tinea corporis, your corpus. Body, anywhere on the body. Tinea manus, on your hands, fingers. Tinea pedis.
Feet.
Yes, exactly, and more commonly called athlete's foot.
Oh, that's a ringworm?
Yeah. And finally, Tinea unguium.
And it's in.
What's an ungulate?
And you're
Hoofed animal. What's a hoof? Modified hair. What are nails? Yeah, so these are the nails. And it's which.
So where they got that?
Where the thing,
Fungus lifts up the toenail and hops on it.
Yeah, and kind of there's this thing that rolls back and forth. Yeah, that's the commercial. And why that commercial? You raise a good point.
So gross.
Because most of these are superficial, correct?
Mm-hmm.
Which means that topicals work quite well to treat these.
But that's why it's a
Tinea,
pill. Or no, is that a pill?
yeah, Tinea unguium is typically under the nail, which means that how easily is this to treat with a topical?
It's not.
Difficult. So you need to take an oral
medication.
antimicrobial or antifungal agent in this case.
But Tinea is also.
Tinea pedis is athlete's foot.
Yeah, and it's hard to.
Okay, what's jock rot?
Is
Women
it
get jock rot just the same that men do. You don't have to wear a jock to get it. It's just chafing in the groin, right? And you got a fungal infection down here. What could that be? Typically it's Tinea corporis, but it can be any of those particular bugs. Kind of an oddball one is subcutaneous mycoses, the third one there, Sporothrix. Look at that word, I just love that word. Lymphocutaneous sporotrichosis. Actually, it's misspelled trichosis.
Mary Poppins.
Yeah, that's funny.
Erase the U out of sporotrichosis. That shouldn't be a U in there. But lympho indicates that somehow lymphatic tissue may get involved in this particular case. Probably everybody in this room has had some infection with a Sporothrix at some time or another. If you had a splinter, and that splinter stayed in for a while and it festered, and what you notice is that when you would kind of pinch the skin where that festering was, you would notice this white kind of dot that appears down below the skin. And that's the mycelium of Sporothrix. Usually what happens is your immune system kicks in and gets it out, or if you're kind of game for it, you can just cut that open and pop that thing out of there. And it looks like one of those goobers you get out of your eyeball, in the morning. You roll around and play with it while you're trying to wake up in class. But if this is allowed to go farther and it ends up in your lymphatic system, this can be deadly. So who's most prone to coming up with a disease like this?
Infants or
Immunocompromised.
adult.
Yeah, somebody who's going to get splinters all the time. And so, interestingly enough, where it shows up most commonly is in florists. So, but it could show up with anybody. Okay, the systemic mycoses. There are a lot to talk about here, and we don't have time for all of them. Are you familiar with any of those 4 that I've listed there? Histoplasmosis, blastomycosis, coccidiomycosis, or cryptococcosis? No. And you heard of any of those? No. Okay, then we'll just deal with one as opposed to making you know all 4. Let's deal with the first one because it's the most common one found around here. So it's called histoplasmosis, Darling disease. Sometimes it's also referred to as Ohio Valley disease. Nobody knows anything about histoplasmosis? Associated in this particular case with certain types of animals.
Like our fish in the river?
Good guess, but wrong.
Oh.
Deer. No, deer.
No, good guess, but wrong.
Raccoons.
Good guess.
Something that we have and no one else has.
No, something we got, but everybody else has got.
Squirrels?
No.
Squirrels.
We find where there are large populations of pigeons or starlings.
Like bird poop?
Yes. And also it is associated with bat guano, but bats, of course, aren't birds. There's usually somebody raises their hands and goes, "They aren't."
Really?
Really, usually there's.
Really?
They fly.
So is this a new thing?
But what's going on here in this particular case is that where you have a lot of, and I don't know if you noticed the flocks of starlings that have still been around here. You know what starlings are, right? These stupid blackish birds that don't belong here.
Right.
They were imported from England by some idiot who thought he wanted to take every bird mentioned in Shakespeare's works and put them in Central Park. Well, where did they end up? All over the country in massive quantities, and they outcompete local birds for nesting, and they're just dirty and disgusting birds, and if you can kill them, get rid of them. Okay, I mean, if the more you can run over on the way home, the better off we are.
That is so funny.
That's my goal for
They are
Hunting starlings.
They're
Hit
getting
massive.
bigger.
But what they do is they harbor the spores of this particular fungi in large quantities, and then when they preen themselves, they swallow those spores, they pass right on through and they poop them out. So there are large quantities of fecal material from starlings and also from rock doves, i.e. pigeons. You know, that fly down, you know, they land all over the place, and people feed them. Why? Why do you feed things that are a nuisance? I don't quite get it.
Right.
So get rid of them too.
Because they're lonely people. Pigeon people. Right.
Oh, I didn't think about that.
Yeah.
Yeah.
Like idiots that don't
Sometimes
have
that's the only friend they've got.
The lady in Mary Poppins.
Yeah.
Feed the birds.
Okay. Sorry about that. Anyway, what goes on with histoplasmosis? So you inhale the spores. And one of the problems with this is it oftentimes gets misdiagnosed. I had a good friend when I was teaching at Thomas More who just began to get sick, had this cough, persistent cough, kept going to his GP. His GP said, "You've got, I think it's walking pneumonia." He gave him all kinds of stuff, and he kept treating him with antibiotics and so on and so on. And he finally got to the point where he couldn't get out of bed anymore. He was so weak. And finally, someone said, "Let's take a sputum sample and send it over to UC Infection Control." And lo and behold, he had these spores in his sputum, and all the time he had histoplasmosis, and he was just misdiagnosed. Oftentimes it's misdiagnosed as either some type of pneumonia or tuberculosis even.
Did they fix it?
Yeah, and he's fine today. But he darn near died because his GP kind of went,
Why wouldn't they have done that in the first place?
I can't be critical of that, but the point was that it was misdiagnosed for a long time, for virtually 2 years. And well, he was skin and bones at the very end. But what does it do? It gets down in your lungs, the mycelium grows in your lungs, It keeps you from respiratory gas exchange properly, and so on and so on. And it can be devastating. It can become systemic, even beyond your lungs, if you give it enough time.
So why him and not us? Like, is where he lived? Or-
And oftentimes, it's tough to say why one person gets it and the other person doesn't. And I don't think we ever knew quite why. He didn't raise pigeons. He didn't spend time in the park. You know, he just happened to get them.
Yeah.
How common is it?
It's we can look on the CDC site. You can look it up and you can come back and report next week. You're in charge.
In Qatar, where my sister lives, that there's so many pigeons, that's where I had heard of that before.
Yeah, exactly.
The workers who clean up the area.
Yeah, and people, oftentimes it's workers,
Yeah.
occupational people, got to come in and clean somebody's attic out, and they've been pigeons living up in there for 1,000 years. Or spelunkers sometimes show up with it. Cavers?
Yeah.
Yeah, bat guano. Bats are not birds. Flip it over. You should recognize a couple of these. Candidiasis.
Thrush.
And so with Candida albicans, and look at the species name, albicans. Alb is the first part of that word. What's an alb?
Like an albatross, like a bird?
No, good guess.
Albino?
Oh, you're on the right track.
So
Your priest wears an alb. It's a white vestment. So it refers to white. So albino, referring to white as well. The same prefix comes out of that. So Candida albicans, typically we think of it in terms of thrush, oral thrush. And if you ever see any kid who's got oral thrush, it looks like he's got cottage cheese in his mouth, where the stuff grows. But it also vaginitis, right? Candidiasis.
I was gonna say, how's the connection? Is it the same thing?
Yeah, same thing. So it just, this particular organism, and everybody in this room, I would guarantee you, I could find a cell in your mouth. Everybody. I guarantee you I can find Candida in your mouth. It's just not uncommon. It's when it outcompetes the other organisms, or the other organisms for some reason are no longer there to compete with it, such as taking antimicrobial medication for an infection in your colon.
Right.
And all of a sudden now we're disrupting, because it goes throughout the entire body, it disrupts the normal flora in your mouth, and you end up with, good case of thrush.
Is that the same situation for C. diff? Like you already have...
It's a similar scenario, that you disrupt the normal flora. And with that then, this bug, which everybody in this room's got in them too, it just outcompetes the others then and takes over. So vaginitis, in many cases attributed to this as well. Aspergillosis, anybody heard of that one? Aspergillus shows up from time to time. The last major problem with it was with peanut butter. Back about 4 or 5. And I still got a jar of that peanut butter that was supposed to have been pulled from the shelf. And this may be 4 or 5 years ago. And I went into my local Kroger store and looked for that VIN number that was, or VIN number that, I was looking for a car in my Kroger store. But the number on the side, which indicates what batches it was, and it was still on their shelf. It was still on their shelf, so I bought it to just bring home. But it was Aspergillus and infected stuff. And what was the problem with it? What would you guess?
Processing the peanuts?
Gastroenteritis in that particular case, because it was in a food product, and a bunch of people got sick. But it also can enter through the lungs as well, and it tends to be kind of an occupational type of a hazard. But there are toxins that are produced by this group, and a very similar group of fungi, and which somebody brought up before about shrooms, and the kind of hallucinogenic kinds of components that are found in certain types of fungi. Hold on, I'll be right back to you. The whole witch thing in Salem, Massachusetts, which we all learned about when we were in the 3rd grade.
I've heard about it.
Yeah. And remember, what was the scenario with that?
They were just all assholes taking
apparently
shrooms, and they
everyone
thought
was-
like, oh, I see this girl floating, she's a witch, but they were just high.
Wow.
That makes me mad. They killed all those people and they were just high.
Maybe.
And there was like ergot growing on the.
Yeah, so ergot. Now, so you're coming up with ergot here, and yeah.
From what
Let's piece this all together. Everybody throw out something
From what
here.
I've read about it, supposedly there was aspergillus on the wheat when they made the bread, and that the people who consumed those batches of bread
They're just
were the
tripping.
ones who were basically tripping their butts off.
Okay, and so anybody else got something to share?
It was rye.
Rye?
Rye.
It was on the grade.
You ate the
It was
rye.
rye bread. Say that again. I like the way you said that.
Rye?
Rye.
He busted out laughing.
Don't ever change. Don't ever change. Don't. Although you can go into the King's English from time to time, correct? So the scenario is this, and most of you kind of hit on it. And about what time period are we talking about? Yeah, the 1600s. And so we got this group of pilgrimish kind of people living in Salem, Massachusetts. And the scenario was, supposedly, that it was a rough summer, there was a lot of rain, and the wheat crop failed miserably, but the rye crop did just fine. But the problem was that the rye, the kernels of grains on the rye, were infected with this aspergillus. And aspergillus is known to produce something called ergot, which some people refer to that as the toxic chemical. Some people refer to it as the abnormal growth of the fungi on there. It gets a little confusing in terms of that. But this chemical, this ergot-based chemical, is very similar to LSD. So the theory still is, although some now have debunked it completely, is that the younger women, whose role it was, some were to be milkers of cows, some were to be bread makers, some were to be seamstresses, whatever it is, that the women who were making the bread were using this ergot-infested rye to make bread for that winter. And they started seeing visions. Wow. And the church fathers with their funny hats on didn't like the idea of the bread makers. If you've ever been to a Grateful Dead concert, there are always the spinners in the back who
That
do
is
this
so
the whole
funny.
concert. The whole concert, they spin, they spin, they spin. But they have a great time. They have a great time. So they were trying to figure out what was going on with the bread makers. And when they saw them supposedly seeing things and doing things and singing inappropriate songs while they made dough, they started saying we need to cleanse them of these evil spirits. And so they drowned them, they burned them, they did all kinds of terrible things to them. Again, at a conference I was at just a couple of years ago, there was a woman from MIT who was there who said this is all just horse pucky. And she went on to show that that wasn't a terrible winter at all, and so on that period of time, and the wheat crop still came in, and so on and so on, and there's no evidence at all that it was ergot poisoning, but it makes a great story. And if you go to Salem, Massachusetts, where they got all these witch thingies, exhibits, you can see that it sells tickets.
It's pretty cool.
And lots of people still go there. So who knows? Who knows? Could it have been true?
Yes.
Yes. But we don't know for sure. Pneumocystis carinii, anybody? What do we know about that? You should all know about this.
That's bad.
What is it associated with? No, these are fungi, quite honestly. It's a respiratory infection.
TB?
No.
Is it?
Okay, as we kind of slow down here today, I'm going to leave that out for you for a little homework. So when you got some time over lunch, though, you can Google that and see what you can learn about pneumocystic pneumoniae, the causative agent Pneumocystis carinii, or you can even use your textbook, and I'll hit you for an answer in lab then. Let's do one other thing real quickly here and before we break. Hello.
I have the thing about bird poop disease.
Bird poop.
Histoplasmosis.
Okay, what have we got to share?
All right, CDC says that between 50 and 80% of people who live in areas where Histoplasma capsulatum is common will show evidence of having been exposed to the fungus. It doesn't say how many people get it though.
Yeah, and that's in the
You have to contact your local health department.
the MMR report actually lists that, which I can get. Yeah, I can pull that down on break. So the question is, you know, I guarantee everybody in this room has been exposed to that fungus. Everybody has. Why don't we come down with the disease? Immune system's intact. You've got enough competition going on with the other bugs that are there. It doesn't pose a problem. Let's do one other thing real quickly here. And just I want to talk about the algae for a second. In just 5 or 10 minutes. And this is a huge group. Well, what do we know about the algae? When you think of algae, you think of water supply, and it could be fresh water, it could be marine water. We use terms like seaweed and all of that, and sometimes seaweed doesn't refer to the algae, it actually refers to a aquatic plant. So it gets very, very confusing out of all of this. The algae, though, are typically put into a series of groups, and it varies between who you're talking about, based on what particular groups of photosynthetic pigments they have. So if they've got chlorophyll A plus this pigment and that pigment, and I'm not asking you to know all of those. It's just that typically we're going to use as a means of classification what specific photosynthetic pigment combinations they have. So we've got things like the green algae, the chlorophytes. And we've got the red algae, which have our rhodophytes. So we start to kind of divide them up in that kind of a way. Why even talk about the algae here? Of what import? Well, from a disease point of view, rarely do we find the algae causing disease, except you go to Florida, you go to South Carolina, North Carolina, and they tell you can't go in the water because there's a red tide.
Lake Erie has that too, don't they?
From time to time. Any body of water, large body of water can have it. A red tide. And you look at it and you go, what's a red tide? Well, you look at the water's edge and it either has this characteristic kind of reddish brown, rust brown color to it. In some cases it can be quite red. And this is due to the presence of a specific type of a algae, which is called a dinoflagellate. And there are a couple of them that are involved, and I'm not going to ask you to know the genus and species of those. That's not important. But what is important is that when these dinoflagellates, obviously flagellated, dino means what? Dinosaur?
Large?
No. No, good guess. Ooh, that was a good guess.
Oh.
No, it refers to armored. Because if you look at these dinoflagellates, they're gorgeous little organisms.
Refers to what?
Armored. And they have a flagellum that runs out that way, another flagellum that runs out this way. It's a single-celled little guy, and he's got these little plates on him of cellulose and derivatives, and so it looks like it's armored. But they produce toxins, and they release these toxins. Now the question is, these are there all the time. Why all of a sudden do we end up with a red tide, and what's the import of that? Something triggers them to reproduce to beat the band. And what's going to typically do that with the algae? I know you're getting tired, so just hold on.
Sunshine.
In the case with anything, things start to reproduce rapidly, get more and more. It's because somebody fed them. And in the case of dinoflagellates, what are they going to be fed? Fertilizer. Fertilizer. So you dump fertilizer on your lawn or on your golf course, and to Hilton Head, you know, how many golf courses are there in Hilton Head?
Too many.
Too many. How many do you need in Hilton Head? Nowhere close to the number we got down there. And how do they keep them green all the time? They dump boatloads of fertilizer on those. Most of that fertilizer never makes it to the soil, but leaches out with a good heavy rain. Where? Into the local creek that goes to the local tributary that goes in the local bays. And you end up with red tides, large fish kills, people getting what appear to be asthma attacks simply walking along the beach, and some people dying if they happen to be stupid enough to go in the water. So we have a toxin involved, and a very potent toxin. So it keeps from going in the water, people from swimming, but more than that, what does it typically affect? Where does the money come in?
Tourism?
I couldn't hear you.
Tourism?
Could be that, yeah, I didn't think of that.
Fish, seafood.
But the other is the whole seafood industry. Because you can't sell any kind of shellfish that are anywhere close to that, because shellfish are filter feeders. They don't get killed by the toxins, but they concentrate them. So now you decide you're going to do a couple of oysters, and you're at the seafood festival in Newport, across the river, and somebody has scooped up a whole boatload of oysters that they got, even though it was a red tide area, you're not supposed to be harboring them, because nobody else had fished there, and it was easy to get them, and claims that they're fresh. They came out of Maine, and you pop a couple of those down, you know, a little bit of Tabasco, and the next morning, you know, you're in anaphylactic shock and you can't breathe and you're dying and you've got the craps that you can't control, and it's because somebody took shellfish from an area that was restricted. Yeah.
I know that we went up to Lake Erie a couple of years ago, and they have big signs all over the place that said, if you see this in the water, don't get in. If you see this in the water,
Yep.
don't get in. And we were on Kelly's Island, and there was actually a gigantic spot that was just red.
Yep.
And it's kind of creepy looking.
Yeah, it is. But the important thing is that they're out there. And these bugs have always been out there, and they're not out to get us. It's just that we've created the condition whereby they can make their way and reproduce to beat the band and simply produce this toxins as a byproduct. Let me throw one other thing out. This will be the last thing. About the algae. What about these? Ever seen those? Heard of those? Diatoms? Unique kind of algae. Kind of a golden brown color. Diatomaceous earth.
Yep.
They're unique little algae. And they come in just a variety of beautiful shapes and forms. Some are round, and they've got all these intricate little markings on their cell walls. They're just gorgeous little things. But their cell walls are made up of silica. So it's a cell wall made up of silica. Actually, in many cases, they look like a petri dish. They've got a lid on the top and a lid on the bottom made out of glass, so on and so on. But a variety of different shapes. Of what import are they?
Are those the ones that are reflective?
No. These are not. Are you talking about being able to fluoresce?
No, like we put in like road paints and stuff like that.
I don't know. Could be. These are typically used when the organism dies. You're left with these little minute glassy particles. It's called diatomaceous earth. It's used for what reason?
To kill bed bugs and other creepy crawlies.
I
If you put them
think
around-
I've heard that there's a use for them there too,
You put
because
it
the
around
bed
your
bugs
baseboards, yeah.
crawl across these little shards of glass, and they cut their little feeties, and they bleed
Cut
to
your feet
death.
off, I'll lame you.
Yeah. Can show up in toothpaste because it's got this gritty kind of a consistency to it, and it scrapes off the biofilm on your teeth. It's also used in filters, pool filters. Loads of uses for these dead diatoms, and there's silica. And silica is kind of glass, right? Unique kinds of cell walls. And there are places around the world where they mine this stuff because they're so thick and millennia ago, more than we could count have died and fallen out of those marine waters and left these huge deposits. But they've got a variety of functions, a whole variety of functions. For the most part, though, what do the algae do? Most of them are kind of free-floating in the ocean. We call them phytoplankton. And more than any of this stuff with diatomaceous earth or toxins produced, what is the overall import of the algae?
Bottom of the food chain.
Exactly right. And being the bottom of the food chain, what do? They have photosynthetic pigments, which means they can photosynthesize. Run with it.
They produce oxygen and allows the rest of life.
Exactly. They produce incredible quantities of oxygen. And at the same time, they take in incredible quantities of carbon dioxide.
They are using real paint.
It is? And I think I'd heard that before, because you get this slight reflection coming off of that, those glassy little particles that are found there. So phytoplankton. Plankton means what? You're simply floating. You're just a floater. So the oceans and the lakes and the streams all across the world are loaded with these little critters whose job it is to photosynthesize, to produce oxygen, but also to do what? If they're at the bottom of the food chain, they're making their own
Glucose.
let's just call them sugars and carbohydrates that simply get passed up the food chain. But right now, even though we see them as playing an extremely important role, what is perhaps the key role for them right now? Reduce atmospheric carbon dioxide. We place so much emphasis on the rainforest as being able to do that, but the algae are much more efficient than the rainforest in terms of doing that. How have we disrupted the algae then?
The CO2 in the atmosphere dissolves into the water and makes the ocean more acidic.
Acidic, exactly.
Does that hurt the algae?
Yes, because it interferes then with enzyme systems that they've got. Small changes in pH can virtually turn off certain enzymes. So then we end up with these phytoplankton no longer functioning at full capacity and not reducing the CO2 in the atmosphere. So it's kind of a vicious cycle that comes out of all of this. They play a key role, and we should thank God every night for the algae. Do you promise me you will?
I'll thank them tonight.
Thank you. Okay, enough for today. You guys get out of here.
We have a test next week.
Yeah, exam next week. What does the exam cover? Let's talk about that real quickly. So that first lecture where we dealt with the major historical events, we came up with, 5 or 6 of them, something like that. Go back and review those. There'll be questions there. I'm not going to ask you specific dates, but I will ask you time periods. General time periods. Did this happen in the 15 and 1600s, or did it happen last year? So I'm not asking you to memorize dates, but just so you get some kind of an idea in terms of that date line that we gave to you and those major events. Take a look at the individuals that we talked about out of that, and what did they do? Just in general, what did they do? Then we got into talking about a little bit about classification, and we talked about Whitaker's kingdom approach and 5-kingdom approach, and then Carl Wose's 3-domain approach. This is all in your book. It's all there. And then we talked a little bit about then trying to classify And we spoke about, in terms of classification, we tried to group things that have what? Have similar genetic makeup. But in some cases, we do it by just comparing morphologies and things like that. And hopefully that the genetics backs that up, and we'll spend really most of the course talking about that, the classification. And then we talked about, and then how do we name things? Remember the scientific notation, a genus name and a species name. And what does that look like? Genus name, first letter capitalized, correct? Underlined. Species name, all lowercase, underlined. And what is a species? A bunch of organisms that
Reproduce.
yeah, but let's just say that genetically they're very similar to one another. Let's keep it simple like that. And then we started to talk about using Wose's kind of three-domain system, talking about the eubacteria. And with that, we said, okay, what are the characteristics of these bacteria? What are the characteristics? And we spent the last lecture talking about a variety of things. We kind of finished that conversation today. And then what did we do? We started talking about some other microbes today. So we talked about the fungi, and we talked about the algae. That's all we got to. And that's all that's going to be on this first exam. Everybody with me? Okay. If you got questions, bring them into lab. We got time to still yak about those. And always come down to my office. I'm right next to the lab down there if you need me.
We're doing lab at 1.
Lab is at 1 today. Okay, so you lab people. Tomorrow's lab also at 1. I'll tell you when I have meetings so that I can't meet at 1. Thanks for reminding me.
It's the morel mushroom.
Oh, morels. Yeah. They're good.
Yeah, they are.
You can't miss those,
Yeah.
so you can't make a mistake.
Expensive. Yeah. $100 an hour. Yeah. $50 something like that. Yeah.
You get big bucks.
Yeah, they're secret spots, but I just found them out at my parents' house. Like the big ash trees, they like to grow around the base of big ash trees on it. It's a bark that comes off.
But then when you
Kind of like
Something that's like the growth of it.
yeah.
There's something in the soil.
Know your tree, know your mushroom.
You're true.
Have you watched, there's a show called Monsters Beside Me? It's from
I've
Animal
had several people talking about that, but I have never seen it.
It's on Netflix.